Let's welcome our next speaker, Dr. Daniel Weinberger. Dr. Weinberger is director and CEO of the LIBD Institute for Brain Development at the Johns Hopkins Medical Center and professor of psychiatry, neurology, neuroscience, and human genetics at the Johns Hopkins School of Medicine. The introduction gets longer and longer. He has been elected to the Institute of Medicine of the National Academy of Science. The title of his talk is Human Brain Transcriptome Dynamics of Neurodevelopmental Disorders. Welcome.
Thank you very much. So thank you. I want to thank Liwei for inviting me to be part of this. This has really just been a spectacular day. I must say I've learned. I mean, my consciousness has been altered from this morning. It's really a fantastic experience. So I'm going to sort of change perspective a little bit and talk about early developmental events related to the later emergence of behavioral syndromes, but I'm going to speak about this in a very molecular context and try to use how genes are regulated in the development of the human brain to look at how they may ultimately represent the origins of some of these later onset behavioral disorders. So there's been a lot of interest in the idea that even adult onset psychiatric disorders have their origins early in development. I think in many ways in schizophrenia, this has become a sort of operational principle. It's generally considered that schizophrenia is a neurodevelopmental disorder. It's not exactly clear what this really means, but this motto was introduced in almost over 25 years ago to explain the clinical observation that schizophrenia, though apparent in late adolescence or early adulthood, occurs in the absence of any evidence of any active neuropathological process around that time. And one of the proposals was that some lesion from early in life interacts with certain normal maturation events that occur much later to lead to the emergence of the syndrome. There's been a lot of circumstantial epidemiological evidence illustrated here based on population studies of early life events that support the idea that the brains of people with schizophrenia don't really develop normally, even though the clinical manifestations that we recognize as this illness don't appear till the second or third decade of life. But there's been no molecular signature of this, but there's been no molecular basis for believing in this hypothesis. So we at the LIBD Institute in Brain Development, before I was there, have tried to argue that we now have some clues to the etiologies of psychiatric illnesses, which are these genes. We've never had any clues to what caused these illnesses. We had a very rich phenomenology, and it keeps getting richer and richer, and we have a very deep descriptive psychopathology. But the first clues we've had to any idea what these illnesses actually are at the cellular and ultimately, if they're developmental level, is from the genes. And schizophrenia, which has become the poster child now for psychiatric genomics, and samples of over 80,000 subjects. This is the work largely from a group around the world. 50 some odd centers that have contributed data to this. There are over 100 loci that have this magical distinction of being GWAS significant. As you know, these are tiny effect size risk factors at the individual subject level. But at the population level, with these huge samples, despite these tiny little effect sizes, there is statistical significance that is basically indisputable as a statistic. The problem is, in all common medical disorders, whether they be schizophrenia, bipolar disorder, anxiety, depression, obesity, asthma, diabetes, heart disease, or stroke, the variants that are associated with these p-values do not affect the structure of a protein. There are much more subtle variations in regulatory elements of the genome, and they affect genes in much more subtle ways than changing the activity of the protein. So how do we translate non-coding variation, which are the underlying genetic origins of these associations? How do we translate these into mechanisms of illness? So at some level, regardless of the regulatory mechanism by which any of these variants affect the biology of a gene, because they're not affecting the protein coding sequence in almost all instances, they have to read out in the transcriptome. And it doesn't matter what mechanism you like, whether you're into epigenetics, or you're into microRNAs, or you're into non-coding RNAs, or looping, or whatever you like. it has to read out in the transcriptome. Now, that doesn't mean you have the right cell, doesn't mean you have the right moment in development of a tissue to see the effect of this, but it has to read out. If you remember from not even at MIT, in grade school biology, there are no recessive genes. There are recessive traits. If you have the right cell and the right moment at which that gene is biologically meaningful, you will see the effect of any variation that impacts on the biology of that gene. So at the Lieber Institute, one of the things we're particularly obsessed with is trying to use the human brain transcriptome across the human lifespan to understand how genes of interest might map onto the developmental programs that build a brain. And this is just to get you in the moods of brains. We have a very large brain project. So we've looked at this now in a number of genes that are of interest because there's some clinical association that links them to the etiology of a psychiatric illness. So let me illustrate this with one of the genes we've been very interested in for a number of years. We reported now almost 5 years ago an association to a potassium channel that's very important in the repolarization of neurons. And we believe this potassium channel, which is the famous hERG channel, critical for the repolarization of heart muscle. But there's a unique isoform of this channel that we discovered, found only in the primate brain, that has a lot to do with the rapid firing characteristics of neurons. Heart muscles cannot fire too rapidly. It's not good for a heart. But brain cells have to fire very rapidly because they have to maintain temporal contingencies over periods of time to allow us to have a memory. And we described that the clinical association that we'd observed to this potassium channel really was about I don't think we have a pointer here, do we? No. Is there a pointer? Okay, great. Thank you. The association that we found to this was to a very specific isoform that we showed only existed in the primate brain, wasn't in the rodent brain. And this specific isoform was very different than the full-length isoform, which had, and this had unique physiological properties and was part of the channelome of the potassium channel biology of rapid firing of neurons. The interesting thing is when we looked at the previously known form of this gene and the genetic association, there was no relationship whatsoever between what was previously thought to be the structure of this gene and the genetic association. Turns out when you look in human brain, it doesn't look like it looks like it doesn't look like the UCSC website, or it doesn't look like the Ensemble website. The human brain has many different forms of genes that are different splice variations, different extended exons, different novel exons, different novel start sites. It's a much different picture when you actually look in the actual brain of a human being than you thought from looking at the public databases. And it turned out that this novel isoform now showed not only association to illness, it showed genetic association to risk association variants, suggesting that this was the molecular mechanism of the association. And then when we looked in normal human brain across the human lifespan, and you're looking here, every dot represents another human being. And these are the prefrontal cortices of independent human beings from 14 weeks of gestational age to almost age 90. And this was the previously known isoform of KCNH2, which obviously became much more abundant during adult life. This was the novel isoform that was associated with risk for schizophrenia. This isoform was much more abundantly expressed during fetal life, suggesting there was something about its regulation that mattered to a developing cortex. And because of the physiological properties of this, we thought, well, if Carla Schatz is right, that neurons that fire together, wire together, this which probably has something to do with the rapid firing characteristics of neurons, might contribute to how these early programs of wiring are developed. So this is one example. I want to give you a few others, because many genes that we've looked at that have various degrees of association with psychiatric illness show this curious property of not only having unique isoforms not previously known, but having a particular propensity to be overexpressed during fetal life. So one of the first genes, this was from the Decode Iceland group, that was ever associated with schizophrenia, which was from a linkage study, is this gene Neuregulin. I show this only because Jerry Fishback, when he was at Harvard, was the person that really put this gene on the map. There was there was a very strong association, a number of early data sets to Neuregulin and schizophrenia, but no one could show that any of the risk-associated sequence variants had anything to do with the known forms of Neuregulin. Turns out that when you looked at Neuregulin in the human brain, there was some previously unknown forms of Neuregulin. And one of the unknown forms of Neuregulin, which became called type IV, had a novel 5' exon that had never previously been identified because, surprisingly enough, people generally studied Neuregulin at the neuromuscular junction. They hadn't really studied it very much in brain. And it turned out that the region of the gene that had shown this prior association in a number of data sets, which had absolutely nothing to do with regulation of the expression of the previously known forms of Neuregulin, now had a strong association to this particular novel isoform. This is just to show that this was a functional variant that we confirmed with a mutagenesis reporter assay. But the bottom line was, now there's a new form of the gene. This is the form of the gene that shows association to the risk region of the genome. And what's also fascinating, this gene is about 4 times more abundant in fetal brain than it was in adult brain. And it's also interesting, this is the only form of Neuregulin that's found only in the brain. It's not found in any other organ. I used to joke around with Jerry Fishback that his worst nightmare with his gene Neuregulin was that no field of medicine would claim it except for psychiatry. That was his worst nightmare, which has absolutely turned out to be true. So we've also seen this, now this is another family of the Neuregulin family, actually the specific ligand for the receptor tyrosine kinase ERB4, which Neuregulin 1 also activates. This is Neuregulin 3. There have been a number of recent studies showing an association with Neuregulin 3 and psychosis. We characterize fully the transcript species of this gene in human brain, and there are many, many isoforms of this gene. Suffice it to say, there's only very specific isoforms that show molecular association to the genetic risk variants. You can't really see this on the axis, but this is 4 times more abundant in fetal brain than adult brain. DISC1, this is one of the most popular genes in psychiatry because it's very penetrant in this family. This was a family from Edinburgh, Scotland, a multi-generational family, where every person that has this arrow had a balanced translocation of the 1st to the 11th chromosome that disrupted several genes, but one of the genes on the 1st chromosome that was disrupted was called disrupted in schizophrenia 1 gene. 2/3 of the people in this pedigree that had this translocation had a major psychiatric illness. They called it schizophrenia because they thought it would get published more easily with that label. But actually, if you look in the families, it didn't really segregate with a particular psychiatric illness. It segregated with a number of psychiatric illnesses, but it clearly was highly penetrant to lead to some psychiatric illness. And nobody could ever show a clear association between risk variants in the gene and any of the known forms of Neuregulin. We did the same old thing, which is because it's become a cottage industry for us. In human brain, we identified about 50 isoforms of DISC1 that were expressed in human brain. And in particular, we identified several very short truncated isoforms, which we showed were made into proteins. And the bottom line was, when we looked at all the known forms of DISC1, none of them were differentially expressed in the brains of people with schizophrenia versus the brains of normal controls. But the novel isoforms were. All these novel short forms of DISC1 were overexpressed, upregulated. We also looked at variants in the gene. These are actually functional variants that Li Wei Tsai has demonstrated biological implications of in tissue. These were 2 SNPs that are functional SNPs in the gene, and both of these show association specifically with these small transcripts and not with any of the previously known transcripts. It's also interesting that these are 2 unlinked genetic variants in the gene showing the same molecular phenotype, which is the principle of allelic heterogeneity, which is probably very common in many genes of interest. People think there's only one royal road to affect the biology of a gene, but through human evolution, there are many ways that you can have the same molecular effect on a gene with different loci within the gene. Lo and behold, This was the previously known form of DISC1. DISC1's been known to be a developmental protein for a long time. It is more abundantly expressed in fetal life, but the novel isoforms are much more abundantly expressed in fetal life. So what about the GWAS data? This was before we had GWAS data. Let me just illustrate two examples from the GWAS data. So GRM3 is a metabotropic glutamate receptor, which is now in the GWAS catalog 10 to the minus 10. I mention this only because this particular SNP right there in the third intron of the gene is right smack in the middle of something we reported back in 2004, which is we looked at GRM3 because it was an obvious gene related to glutamate signaling, and we showed a strong association to a haplotype spanning this particular intron. And there have been many other studies looking at variation in GRM3, but now that it's GWAS positive, it's sort of acceptable. It's been blessed by the broad research community, and now we can study this, even though we studied it a long time ago. But now we can talk about studying it. We don't have to hide it. So the point I want to make is when we looked at GRM3 way back when, this is where all the genetic action is, and we looked at all the known isoforms of GRM3, and we couldn't find any association whatsoever to these molecular isoforms of GRM3, the transcripts, and the genetic variation in the DNA. This was actually the first study we did where we tried to characterize why there was no association between genetic variations here and the known structure of the gene, and we decided to clone GRM3 in human brain tissue to see if maybe the UCSC website didn't really know what was going on in the human brain. And this is an experiment where we try to characterize, we clone transcripts. This is the rapid amplification of cDNA-N strategy. And when we did this and plated what we cloned on a plate, we found that everybody had this band, which was the known structure of GRM3, but there were a number of other bands which didn't map at the molecular weight to any known form of GRM3. When we sequenced these bands, we found that there were obvious variations in GRM3. One of the most interesting ones, by the way, was a variation which was in everyone's brain of a form of the gene that lacked the fourth exon. The reason the fourth exon is interesting is the fourth exon encodes the transmembrane domain of this protein, which means this gene is now encoding a soluble protein. This is a protein that sits in a membrane and transduces in the N-terminus an extracellular signal to a change in intracellular signaling through the C-terminus. But if you don't have a transmembrane domain, this thing is not exactly going to be functioning in this way. And we demonstrated that it's expressed in neurons. It is trafficked to the membrane. It's also in the cytosol. This is doing something in a cell. It's been very poorly characterized since then. But guess what? This is the transcript that the genetic association is linked to. And it's not linked to any previously known transcript. So if you want to understand the relationship between GRM3 and risk for schizophrenia, this is your construct. This is your mouse construct. This is your cell model construct, because this is the molecular association. And here it is again. This is much more abundantly expressed in fetal life. The first genetic association in a GWAS study of schizophrenia was from this study from Mick O'Donovan and colleagues in Wales, and they had an association to a transcription factor called ZNF804A, a zinc finger transcription factor. Nobody knew anything about it. They had no idea still what it does. This is the current data in the PGC2. It hasn't sort of gotten that much stronger, but it's still a GWAS significant signal. We've done the same old thing with CNF804A. This is, we found a very previously uncharacterized, very short transcript, which lacks the first two exons, has this extended third exon, and this is the only transcript that shows association to the risk-associated genotype. Here's the genotype data. This is the common variant genotype that is the risk-associated genotype for schizophrenia. There's decreased expression of this. We've looked at the transcripts. This is a slightly different display, but this is the full length, and this is the novel truncated transcript, which tends to show a pattern of relatively greater fetal expression compared to adult expression. compared to this. So these are examples of individual genes, but we can chip away at this genetic architecture, gene by gene by gene, but this is going to be a very tedious and slow process. And we're doing this, but we also wanted to say, can we take a more global view of the genetic architecture of these illnesses? And one of the ways we've tried to take a global view is to look at the transcriptome based on different categories of genes that are related to neurodevelopmental disorders. And I'm going to show you some results of a recent study that we've done using a data set that we created that is available on our website at the Lieber Institute for Brain Development called Braincloud. I don't know if any of you have gone to Braincloud. This is a wonderful tool that I strongly urge you to explore. This is a tool that's an array-based oligonucleotide expression database. It's not an RNA sequencing database, although we're working on that, and that will be in Braincloud probably within a year. This is on about 300 normal human brains from 14 weeks of gestation to age 90, and you can study the expression patterns of any gene you're interested in, and you can look at genetic variations in the DNA sequence to see how they regulate at different times of life any gene you're interested in. It's a user-friendly graphic user interface. It's a very handy tool. I strongly urge that you look at it. So we were interested in looking at gene sets about putative neurodevelopmental disorders, making a simplistic, low-resolution, first-pass look at gene sets associated with neurodevelopmental disorders, asking a very focused, simplistic, probably unrealistic question, but it was a starting point. And the question was, if a disorder is truly neurodevelopmental, with manifestations from early in life, it is reasonable to hypothesize that this gene might be abundantly expressed in fetal life. And the hypothesis we tested very specifically in this study, and there are many caveats to this. I don't want to over-promote this hypothesis, but it was a first pass. The hypothesis was, are composite gene sets associated with neurodevelopmental disorders preferentially fetally expressed? So we started with 4 disorders that are thought to have neurodevelopmental pathogenesis: schizophrenia, autism spectrum disorder, intellectual disability, and syndromal neurodevelopmental disorders, particularly those that were identified by John Guzella and Michael Trachowski, I'm blocking his name in a moment, in this balanced chromosomal anomaly data set that was published in Cell about 2 years ago. And we also picked 2 other conditions that are less clearly linked to brain development. Bipolar disorder, you could debate how brain developmental it is, but clearly neurodegenerative disorders like Parkinson's disease, Alzheimer's disease, frontotemporal dementia are not generally thought of as neurodevelopmental. And then we looked through the databases for all the evidence that we could acquire from GWAS studies, from CNVs, from sequencing of rare variants, from traditional linkage studies, from functional biological analyses, to identify gene sets that would be associated with any of these disorders. And we asked the question, if we looked in the gene set for the distribution of expression of probes in these genes, would the more neurodevelopmental disorders tend to show greater expression during fetal life than the less neurodevelopmental disorders? So just to give you an example of what we're talking about, here is a gene, ANC3, which is a structural protein that holds a number of ion channels to cell membranes, that is much more abundantly expressed during fetal life than adult life. And this is, again, every dot is all normal human beings in the dorsolateral prefrontal cortex. So here, this is much more abundant. This is a gene, a serotonin receptor gene, the 5-HT2A receptor, much more abundantly expressed, maybe has something to do with the anxiety phenotypes we've heard a lot about later in life. This is our algorithm for looking at the fetal effect. This is a linear regression model where we have a fetal stage, binary variable, fetal life, postnatal life. That's the only question we're asking here. Is it more or less abundant during fetal life than postnatal life, making a gross, oversimplified assumption simply to ask one question. of all the genes linked to any of these disorders, is there a general pattern of there being more fetally abundant than not? We use a surrogate variable analysis in this linear regression to control for a variety of variables that are not necessarily identified, which also includes back variables and aspects related to the processing of post-mortem human brain tissue and RNA. Now, this is a density plot. So if we look at the entire transcriptome as it's represented in Braincloud, which is about 40,000 probes that identify with confidence almost 18,000 annotated genes that are expressed in the prefrontal cortex of human beings. This is sort of the distribution of what across the whole genome, how much genes look to be more fetally abundant than not. So this is this fetal effect is a log2 scale. So 0.5 is about a 1.4-fold greater expression during fetal life. 1 is a 2-fold greater expression during fetal life than adult life. And this is the density plot of all the genes across the genome. As you can see, it shows a nice pattern that about half the genes overall are a little bit more expressed in fetal life than not, etc. So let's look first at the extremes of the developmental spectrum, because if this didn't work at all, we would have stopped this whole project immediately. So we're looking at the extremes of the developmental syndrome. This is the structural, this is the syndromal neurodevelopmental disorders. These are profound developmental disabilities with cranial dysmorphia, dysgenesis, severe intellectual disability. These are the Guzella identified balanced chromosomal abnormality, severe developmental disability syndromes. And as a population, the genes that are 33 genes, he had 40 genes in that paper. We had robust data for 33 of them in Braincloud. This shows a significant tendency for this group of genes to be more expressed during fetal life than adult life. The other thing that was critical was the neurodegeneration genes. 59 genes. This set shows a clear tendency to be less abundantly expressed during fetal life than adult life. I show this because these establish the anchor points for anything that we go beyond now. Because now we're going to move into much less distinct territory with respect to what we know to be neurodevelopmental or not. So let me show you the data for all these different data sets. So most of them don't show a significant difference, but two that do are the Autism Spectrum Disorder Database and the Intellectual Disability Database. Interestingly enough, some of the ones that you might have expected would work, like the CNV database in autism spectrum disorders, didn't. The CNV databases in schizophrenia didn't. There's a trend for the GWAS catalog. This was before the new GWAS data. I'll show you in a second. There was a trend for this. We think the reason these CNV databases doesn't work, and I don't have time to show you these data, is because in these CNV regions, which are very large regions, there are many genes. And the chances are that there are many false positives that are not really contributing to the syndrome of interest in those CNVs. And when we look in the CNV regions, there are obviously a number of genes that do show a strong fetal effect. There are others that don't. We are exploring the possibility that the ones that do may be more relevant to the emergence of these developmental syndromes than the one that don't. Here's the latest data with PGC2. This is 108 GWAS significant loci. Within these loci are about 344 genes. We could map them reliably with the probes on Braincloud to about 287. And there is a trend, a small degree of significance for these genes to show overall a pattern of increased fetal expression compared to adult expression. Again, the thing to remember here is these are catalogs of genes based on p-values. There are likely many genes in these regions that don't relate really to this syndrome, that are passengers in these genomic regions. And so it may be that as we explore in more depth those that do show strong effects, and I show this slide, this is the top 20 genes in the PGC2 data. I just show that for people who like p-values. Because these are great p-values. But they're really, but they just illustrate, again, a p-value and a biological effect are not necessarily the same thing. This is the fetal effect. Again, this is a log 2. So these are pretty strong relative abundance during fetal life. I want to, before ending this, I just want to issue a couple of caveats. This is one region of brain. This is homogeneous tissue. This is an array-based analysis where we're taking probes that are predetermined to identify transcripts of interest. This is a very low resolution strategy for looking at the developmental transcriptome. And also the assumption that for a gene to be relevant to a neurodevelopmental disorder, it should be more abundant in fetal life than postnatal life is an assumption that can easily be challenged. But again, I think as a general characteristic of these gene sets, there seems to be some appreciation of this. I have really nothing to say about environment, even though obviously these genes set the stage for the experience of a postnatal brain, as well as for a prenatal brain. And the other problem in studying schizophrenia is that while we know the environment has to matter, it's actually been very difficult to identify reliable, robust environmental factors that contribute to risk for schizophrenia. Probably the best one is a history of obstetric complications. It's never been clear whether the obstetric complications are because of the genetic problems in development or they contribute to problems in postnatal development. But we did look at obstetric complications in a family study and related them to certain genes that we think are important in mediating the effects of hypoxic ischemic injury. And what this is just to show you that if you looked in families of transmission of alleles in these genes that were picked because of their role in hypoxic ischemic injury, which is thought to have something to do with what obstetric complications mean to a developing brain, inheriting risk-associated variants in the context of obstetric complications had quite a dramatic effect on the risk valence of these genes. And some of these, and I do this in anticipation of Pat Levitt's talk, some of these create a whole new idea of how genes and the environment interact with each other. Because this SNP in BDNF, which is sort of a poster child for tissue growth and development, which changed in combination with a serious obstetric complication, the bias towards risk 12-fold, is a gene in a promoter, is a SNP in a promoter, a functional SNP in a promoter that regulates the expression of BDNF in the placenta, not in the brain, which raises an interesting possibility that this is a way genes and environment interact, having nothing to do directly with the brain, impact brain development because they impact the development of the placenta. So these are the people whose work I've shown you. This is a post-mortem development group, our clinical and molecular genetics groups, and the take-home message is that the molecular mechanisms of association for many putative schizophrenia genes involved altered processing of fetally abundant transcript variants. Gene sets associated with risk for a variety of neurodevelopmental disorders show variable patterns of association with enriched expression during fetal life based on this oligonucleotide array data, and risk association for genes in loci implicated in neurodevelopmental disorders may be improved by conditioning specifically on differential developmental expression patterns. Thank you again for inviting me and for your attention.
Most developmental biologists will tell you that it's very easy to trip up migration and connectivity.
I'm not really surprised, but you know, that's why you do, you know, it's an old saying, if you knew the result, it wouldn't be called research. So that's why you have to do these things. I mean, you know, when I trained in psychiatry, it was across town at a place that doesn't really exist anymore. It was the Massachusetts Mental Health Center. It doesn't exist like it did. And it was a psychoanalytic institution. And we learned that Freud said that what happened to you very in early life mattered. And, you know, he was basically right. Just that he had very few tools to ask these questions at a deeper level. But, you know, I mean, I think we all can appreciate that these early developmental events, probably in many instances fetal events, make a big difference to how a brain adapts to the rest of its life, but to show it.
How many individuals are showing those and yet don't show up with schizophrenia?
Well, we know that's the nature of all common variants. I mean, the problem with all common disorders, it's like many people in this audience have high cholesterol. That doesn't mean they're going to have a heart attack, right? These are risk factors, and the risk factors have to either interact with each other or exist in the context of other risk factors, which are environmental or, or other genes, et cetera. But the nature of common medical disorders is a genetics of being at risk, not a genetics of being ill.